Hello students, this is Neha Chauhan taking biology lecture for class 9. So students, we started with the chapter Fundamental Unit of Life. In that we were discussing about the structure of cell. In structure of cell, we discussed about the boundary of the cell, which is plasma membrane. And in plants, the boundary of cell is composed of plasma membrane plus one additional membrane that is cell wall. Then we discussed about the brain of the cell, which is nucleus. Then after that we were discussing about the cytoplasm. Cytoplasm is the content present between the nucleus and the plasma membrane. So inside the cytoplasm, various organelles are present. So in last class we started with the first organelle, that is the endoplasmic reticulum, which is the factory of the cell and which is also the largest organelle of the cell. So in today's class, we are going to start with the second organelle, which is Golgi body. Pay attention to the pronunciation here. It is Golgi. Golgi bodies, also known as Golgi apparatus, also known as Golgi complex. These Golgi bodies were actually discovered by an Italian scientist, Camillo Golgi, a Nobel laureate. That is, he received a Nobel Prize. He was actually studying on the nervous system of cats. Okay, in that he observed that inside the nerve cells this particular organelle was present, and he named it as, on his name only, he named it as Golgi bodies. Now these Golgi bodies, earlier he thought that these organelles are present only in the nerve cell, but later on it was found out that these organelles are present in all the cells, right? So these organelles discovered by Camillo Golgi are present in all the cells. So in today's class we will be discussing about this organelle Golgi bodies. Right. So in earlier classes, we studied that the endoplasmic reticulum is producing proteins and lipids. Now these proteins and lipids are not used as such by the cell. Rather, these require certain modification, certain editing. Now this modification or editing part is done by a particular organelle, which is Golgi bodies. Now Golgi bodies, let us first look at the structure of Golgi bodies. When we look at the structure of Golgi bodies, in that you see that several tube-like structures, several flat tube-like structures are arranged one above the other, or you can say several flat tube-like structures are arranged parallelly. Okay. Thus forming a whole stack. Now this stack is known as cistern. C-I-S-T-E-R-N. Okay. Now this stack formed by these parallelly placed flat tubes is known as cisterns. On the upper side and lower side of the diagram, you see various round bag-like structures which are actually the vesicles. So this is the structure of Golgi bodies. Let's enter inside a human cell. This is Golgi body taken outside. Now this is the 3D structure of Golgi body. You see vesicles and flat tubules which are arranged parallel to each other in the form of a stack. Now this stack is known as cistern. So this is the 3D structure of Golgi body. So, I repeat, Golgi bodies discovered by Camillo Golgi is present in all the cells, okay, all the eukaryotic cell, and these Golgi bodies are actually responsible for modification of the products formed by the endoplasmic reticulum, and they are composed of flat tube-like structures which are arranged parallel to each other, thus forming a stack. The stack is known as cistern, C-I-S-T-E-R-N, right? So, I told you about the function of Golgi bodies. I told you that whatever is produced by, that is the proteins and lipids produced by the endoplasmic reticulum, are not used as such. Rather, they require certain modification. For the modification, the endoplasmic reticulum sends the proteins and lipids to the Golgi body. Now, how it sends it? So these proteins and lipids, they actually travel from endoplasmic reticulum to the Golgi via bus, or not actually by bus, but by through vesicles, round bag like structures, right? So proteins and lipids from the endoplasmic reticulum, they reach the Golgi bodies via vesicles, round bag like structures. So you can call these vesicles as bus, which are taking the passengers, that is the proteins and lipids, from one place to another, from ER to Golgi, right? So these vesicles, so you you saw the diagram. In the diagram you saw that various vesicles coming, and they coming from the endoplasmic reticulum containing the products, and they were fusing to the flat tubes of the Golgi body, thus transferring the products inside the Golgi body. Now once the product reaches the Golgi body, now Golgi body, what it does is it stores these products. While storing, it modifies. Okay, it modifies these products to make it functional properly, right? And the third thing is, after the modification is done, these products, these modified proteins and lipids are packaged once again. So packaging is the third function. These are packaged once again inside the vesicle, and through the vesicles again they are sent to their target areas, right? So this was all about Golgi bodies, which are the modification center of the cell. Okay, so the products synthesized in the ER are actually modified in the Golgi bodies, and once modified, they are targeted they are sent to their target areas. Right, so I hope you have understood about the second organelle of the cell, which is the modification center of the cell, known as Golgi bodies, Golgi complex, or Golgi apparatus. Right, so after this we are going to start with the third organelle of the cell, which is lysosomes. Okay, lysosomes are actually related to the Golgi bodies. How? Lysosomes are actually synthesized by the Golgi bodies, or you can say that Golgi bodies are involved in the synthesis of lysosomes. Now what are lysosomes? Lysosomes, when you look at them in the diagram, you will see them as round bag like structures. So they are actually similar to the vesicles only, right? They are actually similar to the vesicles only, or you can say that lysosomes are actually specialized type of vesicles, specialized type of round bag like structures, which contain very powerful hydrolytic enzymes, or you can say very powerful enzymes inside it. These enzymes are capable of digesting most of the biomolecules of the cell. Okay, so what is lysosome? Lysosome is formed by the Golgi bodies. Golgi body is involved in the formation of lysosome. They are actually formed by the fusion of the vesicles coming from the lysosome. So lysosomes are actually specialized vesicles containing powerful enzymes which are capable of digesting most of the biomolecules of the cell. And because these enzymes are present inside the membrane, so these enzymes are not exposed to the cell, because if they are exposed to the cell content, then it will digest the whole cell. That is why these enzymes are kept inside the membrane. So you notice this thing, that even the whole cell has a boundary. It has a membrane which separates the content of the cell from the outside cell environment, right? And each and every organelle of the cell, present in each and every organelle of the cell, it also has a membrane. Okay, so each and every organelle of the cell has a membrane. So lysosome also has a membrane which actually separates its content, that is the powerful enzymes, the dangerous enzymes, from the content of the cell, so that the cell is safe and the cell is not digested by those enzymes. Right? Understood. So this is our lysosome. So what is the function of lysosomes? So sometimes what happens is, sometimes there is some damage in the cell. There is some major damage in the cell, and because of that damage, the cell is not able to function properly. Right? So in that case, what happens is, in that case, the lysosome, the membrane of the lysosome bursts open and the enzymes are exposed to the content of the cell. Okay, because the cell is damaged and cell is not functioning properly, so it is better to replace that cell, to digest or remove that cell and replace it with a new cell. So for digesting the old cell, non-functional cell or damaged cell, this lysosome is required. So this is kind of suicide the cell is committing. How? By bursting the membrane of its own lysosome. So this is a kind of suicide done by a cell which is damaged, right? So because of this, lysosomes are known as suicidal bag of the cell. Okay, so this is the first function. Lysosome act as suicidal bags. How? When the cell is damaged, when there is some major damage in the cell, then the membrane of the lysosome bursts open and the contents of the lysosome come out, and that's how it digests the whole cell, and thus makes way for the replacement of the old cell or the damaged cell by a new cell. So is the first function clear? It acts as suicidal bag of the cell. Right. Now the second function. Sometimes what happens is, the damage of cell is not much. Sometimes only a particular organelle is damaged. Particular organelle is damaged, not the whole cell. So it is better to replace that organelle only rather than the whole cell. So in that case, the lysosome, what it does, that damaged organelle, it fuses with the damaged organelle or the organelle which is not functioning properly. So it fuses with that particular organelle and it digests it, and thus makes way for the replacement of that old non-functioning organelle. So here they are acting as cellular housekeepers. Housekeepers means they are responsible for the maintenance of cell. So this is the second function of the lysosome. That is, they act as cellular housekeepers. Right? First function was they act as suicidal bags of the cell when there is some major damage in the cell. When only one organelle is not functioning properly, or you can say when there is some minor damage to the cell, in that case, lysosomes act as cellular housekeepers. Now the third functions. You have studied about how amoeba digests food, takes in food. You have studied the process of endocytosis, in which amoeba forms a cup-like structure from pseudopodia and engulf the food. So amoeba, being a single cell, does not have a separate digestive system. It is a single cell, in that a single cell has to do all the functions. So in that single cell, this lysosome is acting as stomach. This lysosome is acting as intracellular digestive system. Intracellular means, intra means inside the cell. Okay, so in amoeba or unicellular organism, what they do is they act as intracellular digestive system. When the food is coming inside, the vesicle containing food fuses with the lysosome. And once it is fused with the lysosome, the food is digested by the enzymes of lysosome, and the useful materials are absorbed by the cell, and the waste materials are removed out of the cell. So that's how the lysosome functions as intracellular digestive system. So that forms the third function of the lysosome. Now the fourth function. Sometimes the cells may be exposed to some foreign matter. So food is also a foreign matter, but that is useful. Sometimes harmful foreign matter like bacteria and viruses may attack the cell, or they can enter the cell. In that case, what happens is, when they enter the cell, it is the lysosome which digests them, which fuses with the foreign matter, that is bacteria and viruses. It fuses with them, and with the help of its powerful enzymes, it digests the foreign matter or the foreign organisms which are entering the cell, and that forms the fourth function of lysosome, that is protection. So this is about lysosome. Lysosome, its structure, simple structure, round bag-like structure. It is actually a specialized vesicle which contains powerful digestive enzymes, right? And about the function, they act as suicidal bags, they act as cellular housekeepers. They act as intracellular digestive system. Also, they help in protection. So these are the 4 functions of the lysosomes. So in today's class, we have discussed about 2 organelles, that is Golgi complex or Golgi bodies and lysosomes. I hope the function of both are clear. So till now we have discussed about 3 organelles. Endoplasmic reticulum, which is the factory of the cell. It produces proteins and lipids. It is attached to the nucleus. Second, which is Golgi bodies. It is placed near the endoplasmic reticulum. Okay, it acts as modification center of proteins and lipids. And the third thing is lysosome. It has various functions. It contains hydrolytic or powerful enzymes. So till now we should be clear about the structure and functions of these 3 organelles. So I hope you have understood, and I told you that after completing one particular organelle, we are going to add that to our model. So here in the model, I've already added the Golgi body. You can see that this is the Golgi complex, which contains, here I have made 3 parallel flat tubes. This is one tube, second and the third one. Okay, you can see that the vesicles are coming from smooth endoplasmic reticulum. So vesicles are coming from smooth endoplasmic reticulum, and they are fusing here with this flat tube. Okay, this here this is the vesicle which is getting fused with the Golgi body, and once the products, so these vesicles contains products, some proteins and lipids. Now these products, they enter inside the Golgi body. Okay, once they enter inside the Golgi body, the products are modified. After the modifications, the vesicle pinches off from this side. Okay, this is the side facing the nucleus or endoplasmic reticulum. This is the side facing the plasma membrane. So from here, the vesicle pinches off. This vesicle contains the modified product. This pinches off, and the vesicle takes the product to the target area. Right, so you have understood about the Golgi bodies, and this is our lysosomes, the yellow colored lysosomes. Inside it, I have made the hydrolytic enzymes bound by a membrane, thus separating the content of the lysosomes from the content of the cell. Okay, so the boundary of the cell, which is plasma membrane. I have not made cell wall here. So then the brain of the cell, which is nucleus, then the factory of the cell, which is endoplasmic reticulum, then the modification center, that is the Golgi, and then the suicidal bag of the cell, which is lysosome. So that's all for now. I hope it's clear, and that's it for today's class. Thank you.